Usually when we talk about a medical diagnosis, there's this expectation of like total precision. It feels like engineering.
Right, yeah, like it's binary.
Exactly. Yeah. You break your arm, the x-ray shows a jagged white line on the bone, and the doctor just points at the screen and says, there it is.
Yeah, you find comfort in things that are visible, things you can easily categorize.
Right. But then you step into the world of human immunology, and suddenly that x-ray machine is it's completely useless. Yeah. We're looking at a diagnostic landscape that is incredibly murky.
Oh, absolutely. It is the definition of diagnostic muddy waters.
Right. Especially when we start talking about the cells that actually, run the show, the generals of your immune system.
Yeah, the CD4 positive T cells. I mean, for decades, we've known what these cells do, but actually watching them do it in real time, that has been a massive, massive challenge.
Well, welcome to this deep dive. Today, we are unpacking a really groundbreaking May 2026 paper from Nature Communications by Zhang and colleagues. And our mission for you today is to explore this revolutionary new technological platform called APMET CD4.
It's a huge step forward.
It really is. We're going to look closely at how this tool finally allows scientists to track these immune cells in unprecedented detail, and how that is leading to massive discoveries in both COVID-19 and HPV-associated cancers.
And to really understand why this paper is such a turning point, we have to look at those specific cells you mentioned, the CD4 positive T cells.
Okay, so let's define the roles here for everyone listening. If your immune system is, say, a military force, your CD4 T cells are the generals.
Right. They are surveying the battlefield, orchestrating the troops.
Yeah, they send signals to the CD8 killer cells, the foot soldiers doing the actual hand-to-hand combat, and they direct the B cells, which are kind of like the artillery firing off antibodies. But figuring out exactly what these generals are looking at, what specific threats they are reacting to, has basically been a black box.
And the reason it's been so notoriously difficult comes down to the microscopic hardware they use to see the world. So T cells look for threats, which we call antigens.
Right.
Presented on the surface of other cells by molecules called MHC, or major histocompatibility complex.
Okay, so the MHC is basically like a display case on the outside of a cell holding up a piece of a virus saying, hey, I found this, is this bad?
Precisely. Now the foot soldiers, the CD8 cells, they interact with a specific type of display case called MHC class 1.
Okay, class 1.
Right. Think of class 1 like a very tight, closed box. It only holds a really small, rigid 9-amino acid piece of a virus. It's highly predictable.
And because it's rigid, scientists have been able to synthesize it in the lab pretty easily to track those CD8 foot soldiers, right?
Exactly. But the generals, the CD4 cells, they use a totally different display case. They interact with MHC class 2.
And this is where the trouble starts.
Oh, yeah. Class 2 has what biochemists call an open binding pocket. So instead of a closed box, it's more like an open-ended groove. It accommodates these variable length antigens, usually anywhere from 13 to 25 amino acids long.
Okay, wait, so they all share a core sequence.
Yeah, they all have a core 9 amino acid sequence that fits into the groove. But because the ends of the groove are open, the rest of the protein just kind of hangs out over the edges.
So if we look at the interaction between the T cell receptor and the MHC, it's essentially like trying to identify a specific lock, but the key keeps changing its length and shape.
Yes, and because the key keeps changing length, making stable artificial versions of these class 2 molecules in the lab to, fish for specific CD4 cells, it's been a biochemical nightmare.
Because it just falls apart.
Right. When you try to manufacture these complexes so you could observe the T cells, the variable antigen just slips out. The whole thing collapses before you can even use it.
Which brings us to the core invention of this paper, which is the APMET CD4. This is a high-throughput platform that actually solves this physical observation problem. So instead of using those traditional unstable methods, the researchers engineered something called class 2 single-chain trimers, or SCTs.
The molecular engineering of these SCTs is, it's quite elegant. To stop the complex from falling apart, they essentially tied all the separate biological pieces together into one continuous genetic chain.
Wait, how do you even tie molecules together like that?
Well, a natural MHC class 2 molecule has an alpha chain and a beta chain, with the antigen sitting right in the middle. So the researchers took the genetic sequence for the alpha chain, the sequence for the antigen itself, and the sequence for the beta chain, and they physically linked them.
Oh, they use these flexible glycine-rich strings called L1 and L2 linkers, right?
Yeah.
Because glycine is the smallest amino acid, so it acts like a highly flexible molecular hinge.
Exactly. And then To give the entire structure even more stability, they attached a piece called the partial invariant chain, or PII.
Right.
Finally, they added some purification tags, specifically AV tag and His tag on the end. And that allows them to mass-produce these molecules in cell cultures and then easily isolate them using chemical affinity columns.
Okay, I'm picturing this like trying to mass produce synthetic hot dogs for a very specific customer.
Okay, I like where this is going.
The antigen is the hot dog and the MHC is the bun. But because it's a class 2 open pocket, the bun doesn't have ends and the hot dog usually just slides right out onto the floor.
Yeah, it falls right out.
So these researchers basically built a genetically encoded string that ties the hot dog securely to the bun, so it never falls apart when you're using it as bait to fish for those CD4 generals.
That is a brilliant way to look at it, actually. And to prove that this synthetic bait actually works, they started with a baseline test on healthy blood donors.
Just to make sure the bait was functional.
Right. They built a 23-element STT library called CEFT.
Okay.
It's a standard immunological baseline. It checks for memory T cells against common pathogens. We've basically all been exposed to cytomegalovirus, Epstein-Barr, influenza, and tetanus.
And it worked. They successfully caught the specific CD4 T cells looking for those bugs. But what I found totally fascinating was this specific biochemical detail they discovered when they were analyzing why certain antigens worked better than others in these synthetic structures.
Oh, the regression analysis on the stability.
Yeah. Let's do a quick recap here. We have this super stable hot dog and bun system, but they found that the bun naturally favored hot dogs with a very specific chemical makeup, right?
Who did? They found it strongly preferred antigens that have aromatic amino acids positioned in what's called the P1 binding pocket.
Okay, let's break down the physical mechanics of that. When we say aromatic amino acids, we're talking about molecules like phenylalanine, tyrosine, and tryptophan, which, by the way, if you want a mnemonic, biochemists often use the letters F, Y, and W for these.
Right, FYW.
So why do these specific FYW molecules matter so much?
It comes down to their physical shape and chemical properties. Aromatic amino acids have these large, bulky, ring-shaped hydrocarbon structures. And the P1 pocket of the MHC molecule, that central groove we talked about, is deep and hydrophobic, meaning it repels water.
Oh, I see. So when one of these bulky, water-hating rings drops into that deep P1 pocket, it acts like a heavy ship's anchor.
Precisely. It physically wedges itself into the groove, locking the entire antigen sequence tightly in place so it can't slide out. The heavy anchor prevents the hot dog from sliding out of the bun.
That makes perfect sense.
And the reason this finding is so crucial is that it proves the synthetic SCTs are authentically mimicking natural human biology.
Because natural cells do that too.
Exactly. In the human body, natural MHC molecules also heavily rely on those bulky aromatic anchors at the P1 position. It validated that their synthetic lab-made bait was operating under the exact same biochemical rules as a real human cell.
Wow. So we've got this incredibly stable synthetic bait working perfectly in healthy donors. But, you know, the real test for any new immunological tool over the last few years has been the ultimate stress test, COVID-19.
Oh, absolutely. The real-world trial.
Right. They took this tool out of the baseline testing phase and applied it to a dynamic, real-world viral infection.
And their target was the receptor binding domain, or RBD, of the SARS-CoV-2 spike protein. That's the critical piece of the virus that allows it to break into our cells, and it's the primary target for the protective antibodies generated by vaccines.
So they wanted to map exactly which CD4 generals were orchestrating the defense against this specific domain.
Yes, and the sheer scale of the screening here it's just impressive. They didn't just guess which fragments of the RBD might trigger a response. They created a 64-element library that covered the entire RBD in overlapping segments.
It's like taking the protein and slicing it like a loaf of bread, right?
Right.
Making sure the slices overlap so they didn't miss a single possible target sequence.
Exactly. And they used this massive library to screen 50 blood samples from 22 different patients. But, and this is key, there's a vital genetic control they had to implement here.
The HLA type.
Right. All 22 patients in this cohort had to share one specific genetic marker, HLA-DRB101.01.
We should definitely clarify why genetic matching is so important here. The HLA system is basically the genetic blueprint that builds those MHC display cases, or buns, right?
Yes. If you have a group of patients with different HLA types, their bodies are building chemically different display cases.
So a hot dog that fits perfectly in my bun might not fit in yours.
Exactly. By ensuring all 22 patients had the exact same HLA allele, The researchers guaranteed that the CD4 cells across all these different people were looking at the exact same molecular presentation.
Which creates an apples-to-apples comparison. That's brilliant.
And they tracked these matched patients over an incredibly long timeline, from the first week of acute COVID infection all the way out to 36 months post-infection.
3 whole years. And the amount of data they pulled from this is staggering. I mean, for over 2,000 specific CD4 positive T cells, they captured a 6-dimensional profile at a single-cell resolution.
It is a remarkable depth of data. For every single cell they caught, they recorded the antigen specificity, which piece of the virus it was attacking. They recorded the MHC restriction, the patient identity, and the exact time point in the infection.
Whoa, 6 things at once.
Yeah, and the most revealing data points were the TCR alpha and beta genes, giving them the exact genetic blueprint of the cell's receptor and the whole transcriptome.
Let's pause on the transcriptome because that's a term that gets thrown around a lot in biology and we need to unpack it.
Right.
When they capture the whole transcriptome of a single CD4 cell, what does that actually tell us here?
Well, think of the DNA as the complete library of every blueprint a cell has. The transcriptome is the stack of blueprints the cell has actively pulled off the shelf and is using right at that very second.
Oh, okay. So it's the total set of messenger RNA transcripts.
Right. So by reading the transcriptome, you aren't just seeing that it's a CD4 cell. You're reading its internal diary. You can see exactly which genes it is turned on to produce proteins and which ones it is shut down.
The internal diary, I love that. And reading that internal diary allowed them to observe what they call phenotype evolution. They literally watched the behavior and the state of mind of these immune cells change over the course of 3 years.
Exactly. During the acute phase of the infection, when the viral load is peaking and the body is at war, the transcriptomes showed that the CD4 cells were heavily polarized.
It's like pulled in different directions.
Yeah. Some of them looked naive, meaning they were just arriving and hadn't fully specialized yet.
Right.
But many of them were displaying an exhausted-like phenotype.
Meaning their internal diaries were screaming that they were overworked.
Yes. They were upregulating inhibitory receptors, basically hitting their own biological brakes because the continuous viral stimulation was overwhelming them.
But the remarkable part is what happened as the patients moved into recovery. Because months later, as the virus was cleared, those wildly different exhausted cells began to biochemically converge. Their transcriptomes shifted into an effector memory state.
So they calmed down, but they didn't go to sleep. They went into a state of quiet vigilance. They're patrolling the bloodstream, maintaining the physical memory of that specific viral sequence so they could rapidly expand if the virus ever came back.
Okay, mini summary time for you listening. We've got a tool that tracks the generals, watches their diaries, and sees them go from exhausted soldiers to vigilant guards. Now, why does tracking these generals into a memory state matter for the average person going to a pharmacy for, say, a booster shot?
It is the absolute foundation of rational vaccine design.
Okay, how so?
The researchers created an immunogenicity score for every overlapping segment of that viral protein. They ranked the pieces of the virus based on how many unique CD4 clones attacked it, how well those cells expanded, and how long they persisted as memory cells.
And they found a direct correlation, right? The patients whose bodies naturally generated highly immunogenic CD4 responses to specific viral fragments were the exact same patients who had the highest protective B-cell antibody titers in their blood.
Because the generals direct the artillery.
Right.
If a specific viral antigen strongly activates a CD4 cell, that CD4 cell releases chemical signals, cytokines, that instruct the B cells to class switch and pump out massive amounts of high-affinity antibodies.
So if we know exactly which tiny puzzle piece of the virus triggers the best CD4 response, we know exactly which piece we need to put into a vaccine to get the maximum antibody protection.
It takes the trial and error guesswork completely out of the equation.
That's incredible. But, while they were tracking these cells over 3 years, they also stumbled onto a massive clue regarding long COVID. There's a case study in the paper donor ENCOV042 that is it's wild.
It's a very revealing data point. This specific donor suffered from long COVID. And when the researchers used the APMAT CD4 platform to look at this patient's blood 3 years after their initial infection, they found that their SARS-CoV-2 specific CD4 cells hadn't transitioned into that quiet, resting memory state.
Right, they were still actively firing. Their transcriptomes showed they were still in a highly activated effector state, as if the battle was still raging 36 months later.
And physiologically, T cells don't stay in an active effector state unless they are constantly encountering their target.
Meaning the virus was still there.
Strongly suggests it, yes. The fact that these cells are still actively fighting points to the presence of persistent viral antigen. It means that intact virus, or at least fragments of the viral spike protein, are still hiding out somewhere in the patient's tissues.
Like in the gut lining or the nervous system, maybe.
Exactly, continuously triggering the immune response. The generals are still fighting because the enemy is still there.
It's such an incredible insight, and it perfectly bridges the gap between fighting a foreign virus and fighting a threat that has integrated itself into our own biology, which is exactly where the researchers took this technology next.
Right, they transitioned from respiratory viruses to cancer.
Specifically, they focused on patients with precancerous cervical lesions, classified as CIM23, which are driven by the human papillomavirus, HPV-16.
HPV-16 is a master at hijacking cellular machinery. It uses two main oncogenic proteins, E6 and E7, as the engines to drive healthy cells into becoming cancerous tumors.
So since the goal was to find immune cells capable of attacking these specific cancer engines, the researchers scaled up again. They built an even larger 92-element library covering every possible length of those E6 and E7 proteins.
And through this massive screen, they identified 112 specific CD4 positive T cells that were reacting to the cancer proteins. But this is where the paper highlights a massive, often overlooked biological nuance.
Oh, this part blew my mind.
Yeah, that goes back to that open pocket of the MHC class II molecule.
Right, we talked about the core 9 amino acid sequence locking into the deep pocket with those bulky aromatic anchors, the FYW ones. But because the ends of the groove are open, there are extra bits of the protein hanging out over the sides.
Yes. These are called peptide flanking regions, or PFRs.
PFRs, okay.
For a long time, standard immunological models kind of assumed that the core 9 amino acid sequence was the only thing that really mattered for T-cell recognition. But this study conclusively proved that those hanging PFRs dictate the entire functional behavior of the T-cell.
Because the T-cell receptor doesn't just touch the core center of the pocket, does it? Its physical structure drapes over the entire complex, including the edges.
Exactly. And if those flanking regions aren't structurally perfect, the T-cell receptor might still physically bind to the complex, but it won't trigger the intracellular signaling cascade required to actually activate the cell.
Wow. So they demonstrated this by testing 5 specific T-cell receptors they isolated, which they named H1 through H5. And the data on the receptor called H4 is the perfect example of this.
It really is.
When they tested H4 against the synthetic bait, it bound incredibly tightly. If you were just looking at binding affinity, you'd think H4 was a prime candidate to fight the cancer.
But when they put that exact same H4 cell into a live activation assay with actual living cancer cells,
it did absolutely nothing.
It was completely inert. Even though it bound to the target, the slightly different natural flanking regions on the living cancer cell prevented the H4 receptor from initiating an attack.
So it didn't release any cytokines.
Right. It completely failed to secrete killer cytokines like interferon gamma.
And interferon gamma is critical, right? That's the chemical signal that halts viral replication, recruits other immune cells to the tumor site, and can directly induce apoptosis, programmed cell death in the cancer cells.
Yes. If the general binds to the target but doesn't order the cytokine strike, the cancer continues to grow. Binding does not equal functional activation.
Which is a sobering reality for the field of cancer immunotherapy, specifically TCR T-cell therapy.
Let's walk through how TCR T-cell therapy actually works, because it really frames why this distinction is so vital.
Yeah, let's do that.
The idea is to draw blood from a cancer patient, isolate their normal T cells, and use a harmless virus to insert the genetic code for a highly aggressive cancer-specific T-cell receptor into those cells. You then multiply those engineered cells by the billions in a lab and infuse them back into the patient.
So you're essentially building an army of guided missiles custom-tailored to the patient's specific tumor.
Exactly. But if you engineer an army using a receptor like H4, a receptor that binds but doesn't activate, you've infused billions of useless cells. You need a receptor that firmly pulls the trigger.
Okay, so the researchers set up an elite competition among their candidates to find the perfect receptor for human therapy. And out of all the clones, 2 emerged as the absolute frontrunners, H2 and H5.
Both of these receptors showed immense functional activation. They pumped out high levels of interferon gamma and TNF, and in addition, they were incredibly efficient at physically destroying the HPV-driven cancer cells.
But efficacy is only half the battle in immunotherapy, right? The paramount hurdle, the thing that keeps clinical oncologists awake at night, is safety.
Absolutely. Because if you inject billions of highly aggressive engineered T cells into a patient's bloodstream, You have to be absolutely certain they're only going to attack the cancer.
Right. If they accidentally recognize a similar-looking protein on the surface of healthy lung or heart tissue, the therapy could be fatal.
This is why the researchers put H2 and H5 through a grueling, multi-step algorithmic and biological safety test. And they started with a biochemical technique called alanine scanning.
I found this process so fascinating. Okay, so they take the core target sequence of the cancer protein, and one by one, they swap out each individual amino acid for alanine.
Right, and they use alanine because it's chemically neutral. It just has a tiny methyl group, so it removes whatever bulky anchor or electrical charge was at that specific position.
It's a brilliant way to map dependencies. It's like taking a highly complex password and blanking out one character at a time to see which specific keystrokes are absolutely critical for the system to grant access.
That's a great analogy. By seeing which alanine substitutions cause the T cell to stop binding, they mapped the exact molecular fingerprint the H2 and H5 receptors were looking for.
And once they had that exact fingerprint, that exact password, they ran it through a massive blast search against the entire human genome. They were computationally checking to see if any healthy human proteins anywhere in the body accidentally shared that exact password.
But the computational search is only step one. Biology is messy, so they had to prove it in living cells. They took the H2 and H5 receptors and threw them against a panel of 41 different living human cell lines.
And crucially, these cell lines represented 92 distinct HLA types.
They were testing for something called alloreactivity. Since this therapy might be used across a broad population, they had to ensure that the H2 or H5 receptors wouldn't accidentally trigger an attack just because they encountered a different type of HLA bun on a healthy cell.
And the results separated the two frontrunners.
They did. H5, while it was a fantastic killer of cancer cells, showed slight off-target reactivity to one specific HLA type in the healthy cell panel.
Which is a minor cross-reaction, but in clinical therapy, any off-target reactivity is a massive red flag. But what about the H2 receptor?
H2 was completely clean. It showed absolute zero alloreactivity across all 92 HLA types. It successfully recognized and killed cells, presenting the naturally processed, full-length cancer protein without ever touching a single healthy cell.
Wow, mini summary time. H2 emerges from the gauntlet as a massive triumph. It represents a highly potent, perfectly safe candidate that is fully primed for potential clinical translation in human trials.
It's just a phenomenal sequence of scientific problem solving.
It really is. So let's synthesize the big picture of what Zhang and colleagues have achieved here. The APMAT CD4 platform isn't just a niche, theoretical tool for some basement lab. It has fundamentally cleared the muddy waters of human immunology.
For viruses like COVID, we now have a scalable framework for precision vaccine design. We don't have to guess. We can perfectly map the viral pieces that create lasting, highly effective immune memory.
And in the realm of oncology, it solves the daunting needle in a haystack problem of finding safe, highly specific targets for cellular immunotherapies. We can map the entire CD4 response. We can understand the physical nuances of those open pocket flanking regions, the PFRs, and we can isolate therapeutic cells that pull the triggers safely.
It's a paradigm shift in how we observe our own biology.
It really is. But it leaves me with one final lingering thought. A question for you, the listener, to ponder long after you finish listening to this deep dive. We've spent all this time talking about using this incredible tool to track down the exact CD4 generals that are fighting for us, organizing the defense against invading viruses and internal cancers. But what about when the generals turn against us?
Oh, you're talking about autoimmune diseases.
Exactly. Conditions like rheumatoid arthritis, where the immune system attacks our own healthy joints, or multiple sclerosis, where it destroys the myelin sheaths around our nerves. If we can use this exact APMAT CD4 technology to map the precise TCR blueprints of the CD4 cells causing those diseases, could we eventually use this very same tool to find those specific rogue generals?
It completely flips the therapeutic approach. Instead of finding the cells to arm them and multiply them, we map their exact molecular fingerprint so we can develop targeted therapies to induce apoptosis in just those specific rogue clones.
Exactly. You could theoretically surgically remove the cells causing the autoimmune disease while leaving the rest of the patient's immune system perfectly intact. I mean, we started this journey looking for a clean binary X-ray of the immune system. We found that it is deeply complex, heavily dependent on physical context, and constantly evolving.
But with tools like APMAT CD4, the invisible forces orchestrating our biology are finally coming into focus.
They really are. Thank you for joining us on this exploration of the microscopic front lines. Until next time.